Next subtopic for chapter 3, cell division, is 3.4, meiosis. So there are two learning outcomes. So the first one is to describe the stages of meiosis 1 and meiosis 2. The second one is to compare mitosis and meiosis. So first, what is meiosis? So meiosis is a type of nuclear division which produces 4 daughter cells. So the first thing that you have to be able to notice is meiosis will produce 4 daughter cells instead of 2 daughter cells in mitosis. Each of the daughter cells will have half number of chromosomes as the parent cell. Okay, for example, if the parent cell have 2n or diploid number of chromosomes, the daughter cell, the 4 daughter cell will have haploid or n number of chromosomes. So this is what is meant by half. So meiosis occur in reproductive cell of animal which produces sperm or ovum in a process called gametogenesis. So an example of reproductive cell is primordial germ cell. So in plant, meiosis will occur involving cell known as sporocyte. So sporocytes are a group of cells that will produce spore. And there are two types of spore in plants, either megaspore, which is a type of female gametes, or microspore, which is a type of a male gametes. There are two phases of meiosis, which is meiosis 1 or meiosis 2. Remember, when you're writing meiosis 1, you can only use one Roman. Never write it as number 1. Tak boleh. Meiosis 2 pun sama, kena guna huruf Roman. Tak boleh guna nombor 2. Before we go into detail the process of meiosis, there are a few terminologies yang awak kena boleh faham dahulu. So first is sister chromatid. So sama macam proses mitosis, dalam meiosis pun ada structure yang kita panggil sebagai sister chromatids. Okay, similarly, so in meiosis, sister chromatids also refers to the two identical chromatids which are held together by the centromere. Kenapa ada dua identical sister chromatids? Ingat, it's because of the process of DNA synthesis, which occur during the S phase of cell cycle. Okay, so similar to the process of mitosis, before cell enter meiosis, cell tu pun akan ada proses interphase, di mana during interphase tetap berlaku S phase where the DNA will undergo synthesis or DNA replication. That is why untuk proses meiosis pun each chromosome will have two identical chromatids known as sister chromatids, and the sister chromatids are attached to their centromere. Secondly, another term that is exclusive to meiosis is homologous chromosome. So, homologous chromosome is a pair of chromosomes which have the same patterns of genes along the chromosomes, but the nature of the genes may differ. So one member of each pair comes from female parent and the other comes from the male parent. So in this diagram, we can see that each chromosome have its pair. So satu kromosom akan ada pasangan dia yang kita panggil sebagai homologous. So kenapa ada dua kromosom yang mempunyai pattern gene yang lebih kurang sama adalah sebab each of these chromosome came from each parent. For example, in this diagram, the one on the left could come from mother, while the one on the right could come from father. So after undergo replication, each of the chromosome will now have two sister chromatids. The next terminology is bivalent. Bivalent is basically a pair of homologous chromosome next to each other. So macam kita tengok dalam diagram ini, so ada dua homologous chromosomes, the one on the left and the one on the right. And as we can see in this diagram, both of these chromosomes have similar shape and size, as well as similar position of the centromere. Oleh kerana dua kromosom ni ada shape and size yang lebih kurang sama, at the same time position of the centromere pun lebih kurang sama, that's why they are being referred to as a homologous chromosome. Okay, so each of the chromosome bernama homologous chromosome. Oleh kerana both of these homologous chromosomes are sitting next to each other in a pair, at this time they are being referred to as a bivalent. So in this diagram ada satu bivalent. Sometimes bivalent can also be referred to as a tetrad. So another terminology is tetrad. So tetrad is basically refer to the bivalent but specifically to its chromatids. So dalam satu bivalent ada 4 chromatids sebab setiap homologous chromosome ada 2 sister chromatids. So in this case, chromatid nombor 1, nombor 2, nombor 3 dan nombor 4. So bivalent dengan chromatids merujuk kepada pasangan kromosom yang sama. Cuma bivalent lebih kepada sepasang homologous kromosom, tetapi tetrad lebih fokus kepada sepasang bivalent tersebut mengandungi 4 chromatids. So untuk tetrad, tidak semua chromatids tersebut adalah sister ataupun genetically identical. That's why terminologi ini hanya cakap 4 chromatids, bukannya 4 sister chromatids. Only these 2 chromatids are sister sebab dia attached to the same centromere. Sama juga, only these two chromatids are sister sebab dia attached to the same centromere. Kalau kita nak tengok between biru dan merah ini, so 2 chromatids ini bukan sister sebab they are attached to different centromere. Maksudnya they are belongs to different homologous chromosomes. Ingat, setiap homologous chromosome itu are not genetically identical sebab each of them came from different parent. Satu daripada ayah, satu daripada ibu. So dalam terminologi tetrad, kita hanya cakap 4 chromatids, bukannya 4 sister chromatids. Next, synapsis. Synapsis is the process of pairing up of homologous chromosome. So sebelum ni kita dah tengok apakah itu bivalent, iaitu apabila 2 homologous chromosomes form a pair, menjadi sepasang. So proses untuk memasangkan homologous chromosome dan menjadi bivalent, proses itu kita panggil sebagai synapsis. So crossing over is the exchange of genetic material between non-sister chromatids during prophase 1 of meiosis. So crossing over only valid if the genetic material bertukar di antara non-sister chromatids. So kalau kita tengok dalam gambar sebelah kanan ini, so ada kromosom yang color purple, ada kromosom yang bercolor merah. So if the exchange of genetic material occur between the red chromosome and the purple chromosome, barulah kita panggil sebagai crossing over. Kalau in other instance where genes on the red chromosomes exchange to another chromatids of the red chromosome, this is not crossing over because ingat, setiap sister chromatids are genetically identical. So kalau bertukar pun tak akan ada kesan apa-apa. Tetapi if the exchange of gene material occurs between the different chromatids of the homologous chromosomes in a bivalent, so there will be change in the composition of genetic material between the non-sister chromatids of the bivalent. Another terminology is chiasmata. So chiasmata is the region, ingat dia hanyalah kawasan, where crossing over takes place between the non-sister chromatids. So in terms of labeling, we cannot label crossing over because crossing over is a process. Yang kita boleh label adalah chiasmata. So chiasmata is the region where crossing over occur. So back to this diagram. So this is chiasmata, the region where crossing over had occurred. That's why dalam gambar ni kita nampak ada perubahan warna dekat chromatids. Sebab perubahan warna itu nak menunjukkan bahawa sudah berlakunya exchange of genetic materials between the non-sister chromatids. So another diagram to illustrate the process of crossing over. So the one on the left is to illustrate the bivalent. Bivalent because there are two homologous chromosomes sitting next to each other. Because of these two homologous chromosomes sitting next to each other in a bivalent, there is a chance where the genetic material will transfer between the non-sister chromatids. So this is yang kita panggil tadi sebagai chiasmata. So dekat chiasmata ni lah akan berlakunya exchange of genetic material yang kita panggil sebagai crossing over. So kesannya diagram on the right kita nampak ada perubahan warna dekat non-sister chromatid yang sudah berlaku crossing over tadi untuk menunjukkan bahawa sudah ada exchange of genetic material between the non-sister chromatid of the homologous chromosomes. So exchange of genetic materials inilah yang akan menyebabkan berlakunya variation between each daughter compared to the parent cell. So phases of meiosis. So there are two main phases of meiosis, either meiosis 1 or meiosis 2. So each of the phases of meiosis still consists of 4 stages. So nama untuk setiap phases ini sama macam nama untuk stages in mitosis, iaitu ada prophase, metaphase, anaphase and telophase. Cuma kita letak satu atau dua to denote whether the prophase, metaphase, anaphase or telophase occur for meiosis 1 or occur for meiosis 2. Prophase 1 of meiosis 1 is the longest phase of the entire meiosis process. Prophase 1 can further be divided into 5 stages. So there is leptotin, zygotin, pachytin, diplotin and diakinesis. For these 5 stages of prophase 1, you don't have to explain what exactly happens, but you must be able to state the name of these 5 stages and know that these are the 5 stages which occurred during prophase 1. Explanation tak payah, tahu nama pun dah cukup. So what happened during prophase 1? The most important process that happens during prophase 1 is synapsis. So during synapsis, homologous chromosomes will start to pair up to form a bivalent or a tetrad. So because of synapsis, sekarang sudah ada bivalent dan juga tetrad. So because of bivalent or tetrad, it will lead to another process which is crossing over that will occur between the non-sister chromatids. And remember, because of the crossing over, This is why there will be variations in terms of the genetic material between the 4 daughter cells produced after meiosis has completed. So since there is crossing over, there will be also chiasmata formed. So synapsis is the most important process which occurred during prophase 1 that will define the outcome of meiosis in comparison to mitosis. So selain daripada synapsis, other processes that occur during prophase 1 of meiosis 1 that is also similar to prophase of mitosis adalah seperti disintegration of the nuclear envelope. The centrosome starts to migrate to the opposite pole and spindle fiber starts to lengthen. So these three processes sama antara prophase 1 dan juga prophase of mitosis. So the major difference is during prophase 1 ada synapsis, tetapi during prophase of mitosis there is no synapsis. Next is the second stage of meiosis 1, Metaphase 1. Similar to metaphase of mitosis, at this point, the centrosome already arrived at the opposite pole. At the same time, we can see that the chromosomes are now aligned at the metaphase plate. So these two processes are similar between metaphase 1 of meiosis 1 and metaphase of mitosis. But the major difference is metaphase of mitosis, it is the individual chromosomes that align along the metaphase plate. Tetapi untuk metaphase 1 of meiosis 1, the main defining process that differ from metaphase of mitosis is it is the bivalent that arrange or align at the metaphase plate, bukannya individual chromosomes. So kenapa ada bivalent? Sebab semasa prophase 1 tadi sudah berlaku synapsis. So itu the main character that differentiate between metaphase 1 of meiosis 1 and metaphase of mitosis. So another difference that occur during metaphase 1 but did not occur during metaphase of mitosis adalah spindle fiber coming from each centrosome only attached to one of the centromere. So macam kita nampak dalam gambar ni. Okay, so the other centromere will attach to spindle fiber that came from the opposite pole. Kalau dalam mitosis, setiap satu kromosom spindle fiber coming from opposite pole will attach to the same centromere. Another process that occur during metaphase 1 of meiosis that is not the same during metaphase of mitosis. The next stage of meiosis 1 is Anaphase 1. So during Anaphase 1, spindle fiber will start to shorten and pull homologous chromosomes towards the opposite pole. Oleh kerana in previous stage, which is metaphase 1, only one of the centromere attached to the spindle fiber from one pole. That's why yang separated during Anaphase 1 is homologous chromosome, bukan sister chromatids. So during Anaphase 1 of meiosis 1, pasangan homologous chromosome yang terpisah, bukan sister chromatid yang terpisah. So this is why the daughter cell produced during meiosis 1 will become haploid, sebab pasangan homologous chromosome sudah berpisah during Anaphase 1. Next is Telophase 1. So during Telophase 1, the homologous chromosomes which separated during Anaphase 1 already arrive at the opposite pole. At the same time, we can see that the nuclear envelope will start to reappear, surrounding the homologous chromosomes. Next, we can also see that each of the two new daughter nuclei only have half the number of chromosomes when we compare to the parent cell. So during telophase 1 juga, cytokinesis usually akan berlaku. So in animal cell, cytokinesis still occur by the formation of cleavage furrow, but for plant cell, it will occur by formation of cell plate. Sama je macam dalam proses mitosis. So when meiosis 1 has completed, each parent cell will produce 2 daughter cell. Cuma beza dengan mitosis adalah daughter cell produced from meiosis 1 is now haploid, bukannya diploid. Maksudnya, daughter cell selepas meiosis 1 akan ada separuh jumlah kromosom daripada parent cell punya jumlah kromosom. Kalau mitosis, parent dengan daughter cell punya jumlah kromosom akan remain the same. Yang pertama. Yang kedua, because during meiosis 1, specifically prophase 1, berlakunya synapses that involve formation of bivalent. So bivalent pula akan promote crossing over, iaitu exchange of genetic material between the non-sister chromatids. So this is why the daughter cell produced during meiosis 1 is no longer genetically identical, seperti kita nampak dalam gambar rajah ini. So the one on the left, one of the chromatids sudah ada different gene composition. Sama juga dengan homologous chromosome dekat nukleus sebelah kanan. So one of the chromatids pun sudah ada different gene composition. So at this point, walaupun chromatids ini attached to the same centromere, they are no longer genetically identical. Sebab gene dia sudah bertukar dengan the other non-sister chromatids. So basically, selepas habis meiosis 1 pun sudah ada variation in the genetic composition of the daughter cell. Next, selepas habis telophase 1, sel akan terus continue untuk masuk prophase 2 of meiosis 2. So during prophase 2, the nucleoli and the nuclear envelope will start to disappear again. Tadi semasa telophase 1, the nuclear envelope akan starts to reappear. Tetapi masuk sahaja prophase 2, dia akan disappear semula. At the same time, centriole akan starts to move again towards the opposite pole. Sama je macam apa yang berlaku dekat prophase 1 ataupun dekat prophase of mitosis in terms of centriole ataupun movement of the centrosome. Okay, next yang sama juga adalah the formation of new spindle fibers. Okay, notice that tak sama dengan prophase 1. Semasa prophase 2, there is no synapses. There is no bivalent form. There is no crossing over. So, lebih kurang sama dengan apa yang berlaku semasa prophase of mitosis. So, prophase 2 of meiosis 2 lebih similar dengan prophase of mitosis compared to prophase 1 of meiosis 1. Similar, tetap tidak sama. Kenapa tidak sama? Sebab semasa prophase 2, daughter cell ada 2 sekarang. Kalau prophase of mitosis, kita start dengan 1 sel. Kalau prophase 2 of meiosis 2, kita start dengan 2 sel. Itu first difference antara prophase 2 dan juga prophase of mitosis. Second difference is, yang kita nampak dalam gambar ni, kita start dengan sel yang sudah haploid, iaitu dia cuma ada n number of chromosomes. So in this example, each of the daughter cell is n equals to 2. Itu adalah perbezaan kedua antara prophase 2 dengan prophase of mitosis. Perbezaan yang ketiga adalah kita nampak dia punya gene composition di dalam kromosom, iaitu semasa prophase 2 of meiosis 2, the sister chromatids are no longer genetically identical. Tetapi sister chromatid di dalam prophase of mitosis are still genetically identical. Kenapa? Sebab ingat, sel di dalam prophase 2 datang daripada meiosis 1. So semasa meiosis 1 tadi sudah berlaku crossing over, sudah ada exchange of genetic material. So the same cell will continue in meiosis 2. So kalau ada genetic differences between the daughter cell due to meiosis 1, so maksudnya genetic differences tu pun akan carry forward dalam meiosis 2. Next, metaphase 2. So during metaphase 2, the chromosomes ataupun sister chromatids will line up at the metaphase plate. So basically sama macam metaphase of mitosis, di mana individual chromosome will align at the metaphase plate, bukannya bivalent yang align dekat metaphase plate. Okay, so remember, during metaphase 2 of meiosis 2, individual chromosomes align at the metaphase plate, but during metaphase 1 of meiosis 1, it is the bivalent that align on the metaphase plate. The process of metaphase 2 of meiosis 2 is more similar to the process of metaphase of mitosis, sebab dua-dua involve lining up of individual chromosomes. In terms of perbezaan pula, sama macam yang kita mention semasa prophase 1 tadi, iaitu sel akan ada dua. At the same time, sel ini sudah haploid dan bukannya diploid. Yang ketiga, sister chromatids are no longer genetically identical. So itu perbezaan antara process of metaphase 2 of meiosis 2 and metaphase of mitosis. Next, anaphase 2. So during anaphase 2, so at this point, the centromere will divide. So kenapa centromere divide? Is because the spindle fiber will pull the sister chromatids towards the opposite pole. So before the sister chromatid can move to the opposite pole, the centromere will have to split. So kenapa centromere to split? Sebab ditarik oleh spindle fiber. So proses ini lebih kurang dengan apa yang berlaku semasa anaphase of mitosis, iaitu when the spindle fiber shorten, the spindle fiber will pull the centromere, causes the centromere to split. Once the centromere split, the sister chromatid will become separated and starts to move to the opposite pole. So the process of anaphase 2 is also very similar to the process of anaphase of mitosis. Kalau semasa anaphase 1 of meiosis 1 tadi, sister chromatid still not separated because their centromere still not split. So sister chromatid are still attached to the same centromere. So yang separated adalah homologous chromosomes. Sepasang homologous chromosomes yang berada dalam bentuk bivalent tu yang separated. So bila spindle fiber shorten, the homologous chromosome will move to the opposite pole. So the final stage is telophase 2. So during telophase 2, chromosomes will start to uncoil and lengthen. and therefore will start to become invisible again. So, sama juga macam apa yang berlaku semasa telophase of mitosis. At the same time, spindle fiber starts to disappear and nuclear envelope starts to reform again, terbentuk semula. In terms of jumlah kromosom, so the number of chromosome in daughter cell after meiosis 2 has completed remain haploid. Sebab tadi kita start dengan haploid cell, so after meiosis 2 completed pun kita still ada haploid cell. Bukannya bahagi lagi 2. Tak, bahagi 2 tu sekali saja, iaitu semasa meiosis 1. Semasa meiosis 2 tak ada lagi bahagi 2. The number of chromosome ataupun the ploidy number remains the same. Kita start dengan haploid, kita end up pun dengan haploid. Again, at the same time, cytokinesis will occur by formation of cleavage furrow for animal cell or formation of cell plate in plant cell. So this is the products of meiosis 2. Since kita start dengan 2 cell, so each of the original cell will split to form 2 daughter cell. So in total we'll have 4 daughter cell. So each of these daughter cell is haploid and non-genetically identical. Kenapa non-identical? Mainly because of crossing over, which had occurred during prophase 1 of meiosis 1. Importance of meiosis. So there are a few importance of meiosis. So the first one is to produce gametes. Kenapa produce gametes? Sebab gametes mesti ada haploid number of chromosomes. Since meiosis can do that, that's why meiosis involved in production of gametes. Secondly is to produce variation. So the first one is when the haploid cell ataupun gametes fuse during the process of fertilization, there is a recombination of parental genes. Ingat, after meiosis habis, daughter cell, each of the daughter cell are non-genetically identical. So setiap daughter cell tersebut have different gene compositions. So ada pelbagai possibility apabila berlaku fertilization. That's why setiap adik-beradik tidak sama dia punya karakteristik, unless adik-beradik itu adalah kembar seiras, baru sama. Kalau bukan kembar, setiap adik-beradik itu tak akan sama, mainly because of fertilizations of gametes that contain different gene composition. Kenapa different gene composition? Because the cell was produced by meiosis, bukannya mitosis. So another reason for the variation is during metaphase 1, homologous chromosomes separate into daughter cell independently. So another reason is during metaphase 1, homologous chromosomes that align at metaphase plate, they do this independently of each other. So ini kita panggil sebagai independent assortment. Maksudnya, setiap pasangan kromosom ataupun kita panggil sebagai bivalent will align on the metaphase plate independently of the other bivalent. Katakan first pair ini sebelah kiri kromosom dia datang daripada mak, sebelah kanan datang daripada bapa. Yang pasangan di bawah ni pula, sebelah kiri datang pun daripada emak, sebelah kanan pun datang daripada bapa. So ni first possibility. So another possibility is pasangan yang pertama, sebelah kiri emak, sebelah kanan datang daripada bapa, tetapi pasangan yang kedua ini sebelah kiri datang daripada bapa, sebelah kanan datang daripada emak. So apabila homologous chromosome ini separate during anaphase, so maksudnya combination of chromosomes inside the daughter cell will not be the same. Okay, so in the first example, sebelah kiri ada combination of mak dengan mak, sebelah kanan ada bapa dengan bapa. So in the second example, sebelah kiri ada combination of mak dengan bapa, sebelah kanan pun ada combination of mak dengan bapa. So different combination of chromosomes in the daughter cell due to the act of independent assortment. So ini baru dua pasangan. Bayangkan kalau ada 23 pasangan chromosomes di dalam sel tersebut. So lebih banyak different combination of paternal and maternal origin chromosomes. So ini yang dimaksudkan dengan independent assortment. So another reason for variation is the process of crossing over. So ingat, crossing over tadi is referring to the process where there is an exchange of gene material between the non-sister chromatids of a bivalent.